Welcome to my poster presentation entitled Cracking the Black Box of Deep Sequence-Based Protein-Protein Interaction Prediction. My name is Judith Bernett, I'm from TUM, and on the lower left you can find the QR code to the corresponding preprint by me and my co-authors, David Blumenthal from VU in Erlangen and Markus List, also from TUM. While looking at the literature, we found that PPI prediction methods often use only sequence information as input. However, these sequence-based methods report near-perfect results on commonly used datasets, regardless of whether they applied classical machine learning or deep learning. However, few of the methods have source code available and are reproducible, and for the rest, it has not yet been examined systematically whether and how such results are even possible. We investigated 3 scenarios that might explain why sequence-based models can correctly predict whether 2 proteins interact with each other in 3-dimensional space. Now, the first explanation is the one that is favored by the authors of the models, which is that models can detect patterns in the sequences of both proteins that are responsible for determining whether the protein will interact in 3D space, for example, the detection of matching binding sites. Explanations 2 and 3 are both cases of data leakage, which occurs when proteins from the test set occur in training, and you can leverage easier features. Explanation 2 is that the models utilize node-degree information shortcuts that individually explain whether the protein interacts, and then based on these individual tendencies, they predict the interactions. For example, here, we see that P1 only appears in interactions in the training set, so test interactions containing P1 will most likely be predicted as interacting as well. Finally, explanation 3 is that the model merely checks whether the test proteins P1' and P2' are similar or even equal to protein sequences P1 and P2 from the training set, and then predict the label between P1 and P2 for the test interaction. We tested our hypotheses on 7 of the commonly used publicly available datasets. 2 of them are yeast datasets and the rest are human. Because we wanted to investigate the influence of sequence similarities, we downloaded all-against-all similarity networks from CMAP2 for human and yeast. And we used those to partition the proteins for human and yeast into 2 blocks, such that sequence similarity between the blocks is minimized. For our first test, we split the input network randomly, which is the standard procedure used for evaluating state-of-the-art approaches, and we show that this actually introduces data leakage and causes inflated performance measures. We tested 6 deep learning methods, 1 published similarity-based method, SPRINT, 3 similarity-based baseline models, and 2 topology-based baseline models. To investigate the influence of node-degree shortcuts, we randomized the positive input network in the training data by a degree-preserving rewiring, but then we tested on the original test sets. From the performances of the 2 topology-based baseline models, we could also see the influence of learning from node degrees alone. Finally, we tested what happens when neither sequence similarity patterns nor degree shortcuts learned during training can be leveraged at test time, and for that we used the partitioned proteomes. We then split the original datasets such that training was only done on PPIs where both proteins are in P0 and tested them only on PPIs where both proteins are in P1. Also, we could exclusively investigate the influence of sequence similarity leakage by comparing our results against the sequence similarity-based methods. Here are the results of our first test. After random splitting of the original datasets, deep learning and baseline machine learning methods yield interchangeably high results on all datasets caused by the data leakage. We can see that Sprint's AUPRs are as high as the accuracies of the deep learning models on large datasets, which shows that finding similar subsequences to predict PPIs is actually already sufficient to reach excellent performance measures. For our second test case, we see that if we train on biologically meaningless edges but preserve the expected node degree, we do not get random predictions. On the contrary, models can confidently predict PPIs from node degree information shortcuts as performance only decreases slightly compared to the original results. For a third test, we see that excluding training proteins from the test set and minimizing pairwise sequence similarities between training and test strips all methods of their predictive power, When neither node degrees nor sequence similarity is leaked, the methods do not learn any higher level features that could help them with the PPI prediction for unseen proteins. Finally, we tried out what happens if just one protein of the test PPIs is leaked into the training set by training on the cut of the partitions. Here, we again observe strongly elevated performance scores compared to the other setting, which shows that the effect of data leakage is substantial even when just one of the test proteins was seen during training. In summary, we saw that binary PPI predictions are actually already very accurate for proteins seen during training or for proteins that share similar subsequences. High accuracies can therefore be obtained with just simple methods or fast converging machine learning models. We therefore appeal to the community to always compare against baselines and to first try simple methods that cost fewer resources before moving on to complex and deep learning architectures. We also saw that existing methods are actually not equipped for predicting interactions in the dark protein-protein interactome. This was shown by our third test case on unseen proteins. Here we expect that methods leveraging structural information might close this gap in the future. Finally, we think that the extensive training data leakage has actually concealed the full scope of the binary PPI prediction challenge, and therefore, for future machine learning efforts, we provide a large gold standard training, validation, and test set which is free from data leakage and has minimized pairwise sequence similarities. With this, we hope to kindle renewed interest in this machine learning challenge and motivate further progress in the refinement of existing PPI prediction methods. And with that, I want to invite you to check out our preprint and maybe also join our own conference, the Gene Toberfest, which takes place in October in Munich.